presentation titled Investigating a Novel Approach for Treatment of Triple-Negative Breast Cancer.
Thank you, Raihan, for your kind introduction. So, good morning, everyone, and Assalamualaikum. So, as Raihan introduced, I'm Nuramalina Mumin from University of University of Oxford. So, for today's presentation, I'll be sharing with you my ongoing research work, which is to investigate a novel approach for treatment of triple-negative breast cancer. So I'll start off my presentation by answering the question, why is it necessary to do research in cancer? Last week it has been reported on the front page of Brunei Times that cancer is the leading cause of death in Brunei, according to the report by JPKE, and the number of cases has increased like about 13% from 2011 to 2013, from 2011 to 2013. And among the different types of cancer, breast cancer is one of the most common cause of cancer death among women in Brunei. So cancer obviously is one of the major health concerns in Brunei. So there is an urgent need to do research in cancer to have a better understanding about the disease and to improve the treatment for cancer. Breast cancer can be subdivided into three major subclasses for the treatment option. Most breast cancers majority of breast cancer subtypes have a known targeted treatment. For example, the HER2 positive breast cancer patients have a treatment option of a drug called Herceptin, and more than half of all breast cancer patients are progesterone estrogen receptor positive breast cancer, and they have a treatment option of tamoxifen. However, the third subtype of breast cancer, which accounts for approximately 15% of all breast cancer, and it is known as triple-negative breast cancer, and it is a highly aggressive cancer compared to the other subtypes. Unfortunately, for this subtype, there is no known targeted treatment, and the only treatment option is chemotherapy, and the patients tend to have a shorter overall survival after recurrence of the disease. So there is also a need to improve treatment for triple-negative breast cancer. And how do we target cancer cells? So cancer cells differ from normal cells because they receive various signals in the cells, where they have the ability to continuously grow, and they don't have the ability to die, as well as they have the ability to migrate to other parts of the body. And many cancer treatments aims to target one of these characteristics. And in my approach for my research, I aim to target this characteristic simultaneously by targeting a known protein which is involved in this characteristic. It is called HSP90, and the drug is called HSP90. So hopefully by adding the HSP90 inhibitor to the cancer cells, I would be able to prevent the development and progression of cancer cells simultaneously. So my research looks at the HSP90 inhibitor in triple-negative breast cancer, and there are three major aims in my project. So the first aim is to look at the activity of the drug in the triple-negative breast cancer, whether it is able to have any effect in the breast cancer. And the next aim is to investigate whether there are any potential treatment marker for the drug. This might be helpful for predicting individual patient response to the treatment. And the next aim is to find out whether there there is any potential drug combination to increase the efficiency of the treatment. So I'll be explaining the experimental approach that I've used in my project. So basically, I've used stable cancer cells derived from breast cancer patients, and these cells are grown outside the body, and several experiments performed on these cells, for example, genetic profiling and multi-drug screening. And first, I'll share with you to find out whether the cells, the triple-negative breast cancer cells, are actually sensitive to the treatment. So the video on the right, I'm going to play later, so I'm just going to explain it first. So the video on my right, the cancer cells that were untreated, so meaning there weren't any drug present, whereas the video on my my right is the cancer cells that were treated with the drug. So basically, over time, in the untreated cells, the cells appear to be happily growing, whereas in the treated cells, you can see there's an increase in white spots. So basically, they are the dead cells, meaning that the treatment of the drug was effective in killing the cancer cells. And I've also performed other experiments, for example, cell proliferation assay, Western blot flow cytometry. I'm not going to explain them in detail now, but I'm more than happy to share that later on if you're interested to find out more about it. But basically what I can conclude from this slide is that the triple-negative breast cancer cells that I've used in my experiment were sensitive to the drug treatment, and I was able to proceed to the major experiment, which is genetic profiling. So genetic profiling. In this experiment, I've treated the cells with the drug and took out their RNA, and after that sent them to next generation sequencing. And next generation sequencing is the leading technology for sequencing genetic materials, and you're able to look at the expression of genes simultaneously, like a whole lot of genes simultaneously. This project is still ongoing, so I can't really show my results, but I can explain to you all what sort of results that I kind of expect from this experiment. So the result will look something similar like this, and from this plot, you'll be able to find which genes we can see from the plot which genes are showing increased expression or decrease in expression after the treatment of the drug. It is more like fingerprinting of cancer cells, because cancer cells differ from each other as well, and they respond differently to the treatment. So hopefully, from this experiment, I hope to find like whether there are any genes that are able to predict the response to the treatment, and it could potentially become a treatment marker to predict individual response to the treatment. The next approach that I've used in my experiment, and the next approach that I've used in my project is multi-drug screening. In this experiment, I've looked at the sensitivity of about 326 drugs in combination with the HSP90 inhibitor. In this experiment, I've performed an experimental assay to detect the number of cells left in the plate after the treatment. So if there were less cells left after the treatment, it suggested that cells were sensitive to the drug combination, meaning that it was effective. However, if there were more cells after the treatment, it just explains that the drug combination was not good. So this experiment is also ongoing, so I can't really show the result. But I hope that from this experiment, I will be able to find any potential drug combination and target, and possibly a new target for treating triple-negative breast cancer. So I've reached my conclusion slide. I hope that I've showed that cancer research is necessary, and there is a need for finding new treatment and improving the treatment for triple-negative breast cancer. And I've also mentioned an approach of using HSP90 inhibitor in triple-negative breast cancer. And I've listed down the possible impact of my research on cancer research as well as to Brunei. So hopefully by having a greater understanding of the drug may help in the development of treatment for triple-negative breast cancer, as well as it may also help in the clinical management of cancer patients in the long run by predicting individual patient response to the treatment. And also from the drug combination, there is a potential for developing new drugs and new targets for triple-negative breast cancer. And this impact can also possibly help in realizing Brunei Vision 2035, which is towards a healthy nation, and also hopefully it can contribute to a dynamic and sustainable economy. And I would like to take this opportunity to thank His Majesty, the Sultan of Brunei, and his government for funding my studies, as well as University Brunei Darussalam for the scholarship, as well as University of Oxford for providing a place for me to do this research. As well, thank you to both my supervisor, Dr. Anderson Ryan and Dr. Anthony Kong, for their constant supervision and support, patience, as well as my lab members from both research groups. And also, I'd like to thank you all for your time. And also, I hope that by sharing my research, at least I will be able to inspire anyone in the audience to contribute to creative research, as what this symposium team suggests. Yeah. Thank you.
Thank you, Dayangku, Nuramalina, and we now welcome the audience to direct questions if they wish to our exhibition presenter.
Hi, good morning. It's great to see the presentation. I just like to ask perhaps a bit more of the background for the audience to understand. HSP90, is it expressed in other normal cells besides just breast cancer? And if you use this drug, would it also affect the other cells, the cancer cells? How toxic is it?
So basically, just a little bit more background about HSP90 inhibitor. So HSP90 inhibitor is a molecular chaperone protein. It is known to help in the folding and maturation of proteins. So not only, how to say this, proteins in the normal cells, as well as, not only in the cancer cells, but also in the normal cells. However, there's also several studies that have been shown that this HSP90 has a higher dependency towards the oncogenic proteins compared to the normal cells. So there's always a problem with targeting proteins, because obviously they're both expressed in the normal and in the cancer cells. However, HSP90 inhibitors known to target HSP90 expressed in cancer cells. There's a bit of a higher binding affinity to the HSP90 expressed in cancer cells. Yes. Yeah. I hope I've answered that question. Yeah.
Assalamu alaikum. Thank you very much for the informative speech. Although it's far less common, I would like to know your thoughts on breast cancer in men. Are there any differences in the cancer cell structure? What are the symptoms, diagnosis, and where can we find more information? Thank you.
Obviously, breast cancer is not only detected in women, it also can be found in men. I mean, it's rarely, but it's not how to say, impossible. But I don't think so. I have that knowledge to compare what's the difference between the breast cancer, because obviously, maybe someone in the audience, I can see Fatin is raising her hand, so maybe she can answer more on that. Sorry, I don't think so I have that.
Just to answer that question, I'm not really sure, but I've read somewhere that breast cancer can happen in men, and if it happens in men, it will be more invasive, and it will be more worse, the symptoms will be more worse, and it will be harder to treat. Yeah, I think it will be harder to if it happens in men. Yeah, just from my general opinion, I think so.
I have a question. When you return to Brunei, do you intend to continue your research on cancer cells? And if you do, how do you intend to do that with the resources that are only available in Brunei and your research partners that are only available in Brunei?
So that's a very good question because I have a strong passion in doing research, and that's why I've decided to do a DPhil in oncology as well. So before me, I'm not sure if I should give a whole background about myself, I guess. So my scholarship is from University Brunei Darussalam, and so I'm already affiliated with the university. And also before I continued my studies, I did a research. I was working as a research assistant in UBD, so at the Institute of Health Sciences. So I'm kind of familiar with the facilities available there. So they have their own tissue culture room, as well as they have a how to say, a sequencer, but they're not like the next generation sequencing, but then there's how to say, there's a sequencer, first generation sequencer, and then there's also microscope. So hopefully I'll be able to do more research and continue my research in UBD at the Institute of Health Sciences.
We'll take one last question before we proceed to the next sequencing event.
I was just wondering, what's the difference between the 3 different types of cancer you had on your pie chart, and also this HSP90? It sounds like it attacks like a very general protein, right? So does it have the potential to actually cure every other kind of cancer, or is it specifically for breast cancer?
So I actually forgot to mention that. So the 3 different subtypes of breast cancer. There are several ways of subclassifying breast cancer. The one that I've shown is based for their treatment option, because there's the 3 major options, whether they're protein, like targeted treatment, or hormonal treatment. So the progesterone estrogen receptors, they're hormone receptors, so that's why they're a hormone-based treatment called tamoxifen. Whereas the HER2-positive breast cancer, it's a HER2 overexpressing protein in breast cancer patients. So the treatment option would be Herceptin, which targets the HER2 protein. And the subtype that I'm working on, which is a triple-negative breast cancer, obviously it's negative, meaning that they don't express HER2 at all, and they don't express progesterone estrogen receptors. So that's why they don't really have a targeted treatment. So in terms of HSP90 inhibitor, it's not only in breast cancer, it's actually, there are a lot of HSP90 inhibitors in the clinical trials, not only in breast, but as well in lung cancer, and it is currently in phase 3 clinical trials, so in lung cancer, but not in breast cancer. But yeah, breast cancer maybe it's only phase 2 clinical trials. And there are several HSP90 inhibitors, and the one that I'm working on, because I don't want to put up like a term there, so it's actually I'm working on a drug called Ganitaspib. So there's like Ganitaspib, 17-AAG, there's so many names, I guess. So yeah.
Thank you very much, Dayangku Nuramalina, for your presentation.